our second video on the chapter of the central nervous system, and it's about the electrical activity of the central nervous system. This video is divided into three segments. First off, we will start with the resting membrane potential, then the local potential, and in the third segment we will discuss the process of action potential. In this video we will be using this graph and taking millivolts and milliseconds as our units of measurement. The resting membrane potential is a feature of all body cells, and it's the difference in electrical potential between the inside and outside of the cell membrane when the cell is at rest. The cell membrane has a negative charge from the inside, and we say that the cell is polarized, and it has a positive charge on the outside. The electrical potential is measured by a device called cathode ray oscilloscope by placing two electrodes, one on the inside of the cell membrane and the other on the outside of the cell membrane, and it measures the difference in electrical potential. Regarding ionic distribution, the inside of the cell has a higher concentration of potassium and phosphate, while sodium chloride and calcium are more concentrated on the outside of the cell. There are three factors causing ionic movement across the cell membrane, and these factors are the concentration gradient, the electrical gradient, and the permeability of the cell membrane for the ion. Here we are taking potassium ion as an example. Potassium concentration in the intracellular fluid is about 140 millimole per liter, while on the extracellular fluid it's about 40 millimole per liter. And for that, under concentration gradient, potassium will move to the outside of the cell. But remember that the cell membrane has a positive charge on the outside, and this positive charge will push back the potassium to the inside of the cell. The cell membrane has a potassium channels that transports the potassium freely across the cell membrane. The potassium ion will move across the cell membrane until reaching an equilibrium potential, in which the chemical gradient is equal to the electrical gradient, and the equilibrium potential for the potassium is about -90 millivolt. Nernst equation is used to calculate the equilibrium potential for a specific ion. Now we move on into the causes of the resting membrane potential and why the cell membrane is negative from the inside. There are three factors that contribute to the resting membrane potential. First off, the presence of potassium leak channels, which freely transports potassium to the outside of the cell through its concentration gradient, and thus the cell is losing a positive charge which will be transported to the outside of the cell. And this is the most important factor in the genesis of the resting membrane potential. Second cause is the presence of sodium-potassium ATPase pump, transporting 3 sodium ions into the outside of the cell and 2 potassium ions to the inside, against their concentration gradient with every activity. And the third cause being the presence of anions like proteins and organic phosphates that could not be transported to the outside of the cell due to its large size, and it has a minor contribution on the genesis of the resting membrane potential. The resting membrane potential is measured using the Goldman's equation, and it's about -70 millivolts in the neuronal cells. So that's it for the resting membrane potential. Now let's move on to the local potential. And now with the second segment of this video, we will go through the local potential. The local potential arises when there is a movement of charge causing a change in electrical potential, but it is limited to a particular site on the cell membrane, typically about 1 to 2 millimeters from the point of origin. The local potential occurs on the parts of the cell membrane receiving signals, whether it was the dendrites, the cell body, the environmental sensory receptors or motor end plate. The local potential is caused by a minimal ionic movement not reaching the firing level or the threshold, a point in which an action potential takes place, and it's about minus 55 millivolts for the neuronal cell. For example, this neuron is receiving signals through its receptors, which are connected to a chemical gated sodium channel. The result is a subthreshold partial depolarization. The local potential is graded to the strength of the stimulus. So the stronger the stimulus, the larger the amplitude. Summation of electrical charges is possible on the local potential, meaning that if two receptors are excited at the same time or one receptor had two different stimuli, the strength of the two stimuli's amplitude will be summated in the genesis of the local potential. So that's it for the local potential. Now let's move on to the last segment of this video about the action potential. And then with the last segment of this video, the action potential. Action potentials occurs when there is a strong stimulus reaching the firing level or the threshold. Once the threshold is reached, a massive opening of voltage-gated sodium channels occurs, causes sodium ions to go to the inside of the cell, down its concentration gradient, and this process is called the sodium influx. The sodium ions are trying to reach their equilibrium potential, which is about positive 60 millivolts. But that does not happen because these channels are fast to open and fast to close. This process is called depolarization because the cell is losing its negative charge or its negative polarity. The voltage-gated sodium channels goes into an inactive state before completely shutting down. And at this point, there will be a massive opening of voltage-gated potassium channels, causing the potassium ions to go to the outside of the cell down its concentration gradient, a process called potassium efflux. The cell is gaining back its negative polarity, and thus this process is called repolarization. Because potassium voltage-gated channels are slow to open and to close, the cell membrane will reach a state of hyperpolarization. Now the process of action potential will be repeated over and over, and it will be propagated through the whole length of the axon. So let's look at the process of action potential on our graph. First off, a stimulus reaching the threshold potential, and the period from the stimulus initiation to the threshold potential is called the latent period. Then depolarization occurs, and when the cell membrane's electrical potential becomes positive, we call it a period of overshooting. And then repolarization occurs, and when it's about 70% completed, there will be a slower rate of repolarization, a period called the after depolarization, in which most of the sodium voltage-gated channels are completely shut down. Then a state of hyperpolarization, and this last segment is called the after hyperpolarization. So this is about the process of action potential. Now this process has three characteristics. So let's move on to check out these characteristics. Now let's look at an important characteristics that differentiates the action potential from other electrical activities. First off, the all or none law. For the process of action potential to occur, the strength of the stimulus must reach the threshold or the firing level, and from there the process is initiated. So it's either there is an action potential or there is no action potential. There are no in-betweens. The second characteristic of the action potential is the refractory period, a period in which the cell membrane is refractory to excitation, and it's divided into an absolute refractory period in which no stimulus, no matter how strong is that stimulus, could affect the cell's electrical potential. It's the period from the threshold potential till third of the repolarization is completed. So here in blue, representing the absolute refractory period. From the end of the absolute refractory period till the start of the after depolarization is the relative refractory period, in which only a strong suprathreshold potential could excite the cell. Represented here in green color, the relative refractory period. Then the third characteristic of action potential is the conduction or propagation. The action potential is an active self-propagating process with no change in amplitude or velocity. So when it starts at a point, it will be propagated through the whole length of the axon. There are two types of conduction according to the type of nerve fibers. First off, the saltatory conduction, and this takes place in the myelinated nerve fibers, in which no action potential occurs in the myelinated areas. Action potential only happens on the nodes of Ranvier, jumping through the myelinated sides. This type has one direction of propagation, only forward propagation, and it's characterized by fast conduction. Second type is the continuous conduction, taking place in unmyelinated nerve fibers, characterized by circular movement of current and the two direction of propagation, forward and backward propagation, and it's much slower than the saltatory propagation. Now, before we are done with this video, there are three important factors that determine the velocity of the conduction in the nerve fiber. First off, the degree of myelination. The thick myelinated fibers conducts electricity faster than thin myelinated fibers, and the thin myelinated fibers conducts faster than non-myelinated fibers. Second is the distance between the nodes of Ranvier. The longer the distance between the nodes of Ranvier, the faster the conduction velocity. And then the last factor determining the conduction velocity is the axonal diameter. The larger the diameter of the axon, the faster the conduction velocity. So that's it guys for this video. Thanks for watching and see you in the next video.